great to see lots of familiar faces. Thank you for coming to the third meet and greet of the Global Frugal Diagnostics Network. We're always excited about the turnout and your enthusiasm for building this community. I just wanted to very quickly introduce myself. My name is Ayo Kunle-Olarinwaju. I'm an assistant professor at the University of Washington in bioengineering and mechanical engineering. Technically, my background is in microfluidics, capillary-driven microfluidics, and I also do enzymatic reactions for HIV drug monitoring. But on a more personal note, part of my interest in wanting this group to exist and wanting to collaborate with a lot of you is I grew up in Nigeria. I grew up in the southwest of Nigeria. I did a lot of my training there in a small town called Chagamu. And there were two things there. There was a hospital and a cement factory. And a lot of the people I knew were either doctors, nurses, or they were engineers. And I've always been trying to be in the middle of those two places. And then as life would have it, I moved to Canada, to Fort St. John, British Columbia, on the Alaska Highway when I was 16. And I remember back then, I thought I was going to St. John's, Newfoundland, near the Atlantic, which would feel familiar. And I ended up sort of in the middle of nowhere. It was especially wintry. And I think, again, this sort of dual experience of living in two different places is also kind of provided for me this importance of what exists in one place and what exists in the other, and how do we bring those together? And in what ways are those complementary? In what ways are we building something for here and building something for there? And in some ways, in what ways is global health also local health? Is what's local to here, local to there? So for me, there's this excitement and enthusiasm to think about how, as academics, what we do can translate between spaces. I think what I'm especially excited about, and I think what I hope that our presentations today showcase, is showcasing work that happens in many different places, right? And showcasing work that allows us to build capacity collectively and build community collectively. And we really hope that as these virtual and in-person meetings continue, we can show more examples of work that you all are doing in different places, and that really informs and builds all of us together. So enough about me, you're not here to see me. This is what we're talking about today. So we're going to very briefly recap what the network's goals are and our previous meetings, because I know some of you might not have been here in the past, or maybe it's been a few months and it's all kind of foggy. This will be very, very quick. We have two presentations today. We're going to have a very short flash talk by Brian Matovu from Makerere University at Uganda. And he's going to talk to us about some of the early stages of prototyping microfluidics. And our hope is really that this forum can also provide a way for people to show work in progress, where we get feedback on some of the things that they want to do in their communities. And really our hope is that others in the community will reach out and basically try and present like this. So if you're interested, reach out to us in Slack, by email, we can talk about it and involve more of us, because we really want to showcase sort of the global reach and this global network. Then we'll have a presentation by Dr. Manu Prakash from Stanford University about capacity building via community-driven science. I'll do a more detailed introduction to that later on, but I just wanted to recap who we are, why we're here, our objectives, our activities. So we're a community of researchers and practitioners working and hoping to create an equitable, user-led, decentralized, and sustainable diagnostics innovation ecosystem in global health. And really, our hope is that this involves all of us. We're focused on R&D for global health diagnostics. We want to share, transfer knowledge, share innovations, good practices, democratize access to diagnostics. And we're doing a number of online workshops. You're here at one. We want to provide toolkits and resources and develop frameworks. This is just a very, very brief summary of what we've done so far. So we've had 2 of these online meet and greets so far. We've got blog posts for both of them. Dr. Alice Street at the University of Edinburgh kindly wrote those online. And we've also got a recording of the second one. So if you missed any of the things we did in the past, please join us. What we're especially excited about, though, is you, the great turnout. How many of you show this enthusiasm and interest? And it's really great to see the range of countries and the number of participants. And we hope that we can continue to grow and expand, because we know a lot of this has been word of mouth. So if you know people who work in this space or active in this space, please reach out to them and involve them. We want all of you to be involved. With that said, we're going to have our first flash talk, and I'm going to hand over to Dr. Kersaudy-Kerhoas for a quick introduction.
Thank you very much, Ayo. Good morning or good afternoon to all of you. So I'm Kersaudy-Kerhoas. I'm a professor of microfluidic engineering at Hertford University in Edinburgh, Scotland. And together with Ayo, Alice, Elena, and Harry, and Z, I've founded the Global Frugal Diagnostic Network. In my practice, I try to develop prototyping technology for microfluidics, and I try to make these prototyping technologies really accessible so they can be deployed by people all over the world. I'm delighted today to chair this flash talk by Brian Matovu from Makerere University in Uganda. Makerere is a university really known for its biomedical engineering work, and you might have heard about their work. They also have a strong Scottish connection because one of the principal investigators there has a PhD from a Scottish university. So Brian and I actually connected via social media and online forums, and most recently via one of his students on the Reclone platform. And some people might join from this different community. And I'm really glad that Brian and the biomedical engineering team at Makerere have joined the network. So I've invited them to really share their progress with microfluidic prototyping. And I really believe that, their efforts shows that, simple tools can enable technology demonstration and R&D. And when I was talking on the Reclone platform, Edgar, who was there from Makerere, said, "Oh, but I can't produce these prototypes," and I bet that he could. And I met him the next day, to talk about microfluidic prototyping. And here we are, just a month in or a few weeks in, and Brian is able to present this progress. So over to you, Brian.
Right. Thank you, Prof. Maywin and Prof. Ayo for the introductions. So once again, I'm Brian Matovu from Makerere University. I am a biomedical engineering researcher and innovator at the biomedical engineering unit of Makerere University. So I'm here to share briefly our capabilities, okay, and testing our potential to take on the microfluidic diagnostics in Uganda, especially at Makerere University. So Makerere University is one of the oldest universities in the region and the oldest in Uganda. And last year, we just celebrated 100 years of existence since 1922. But within that, the biomedical engineering unit, which is now celebrating around 12 years of existence, we have a vision of being the leading center of biomedical engineering academics, research, and innovation excellence in Africa. So focusing on design, through also one of our collaborations with Duke University, we were able to set up a design workshop, okay, or a design center where students and faculty can actually prototype and come up with bring their ideas to reality. And this was made from 2 shipping containers that were put together and equipped with the necessary tools that the students use and faculty use. And also a similar model was put at Duke University in the USA to put the idea of using the shipping containers, putting them to use. And still focusing on that, we've been able to do prototyping for a different, so many other innovations. And we really haven't been having much of innovations around diagnostics. So for example, we have the early preeclampsia diagnostic strip and for diagnosing preeclampsia in pregnant women. So at this stage, we are, I think, soon going in for trials. We also have Lumenda for diagnosing meningitis. So these are the few diagnostic innovations that we've had at the biomedical engineering unit. In Makerere, through the other colleges and departments, there are a few other diagnostics, but specific to us, I think we've only had those 2. However, we've had so many other innovations like the low back corset. We have the Kersaudy device for laparoscopic surgeries. We have the gastroenteritis bags for the babies who are born with their intestines out. And all these are being supported through this design container, which is equipped with necessary tools from electronics and a few mechanical tools that the students can use and faculty, and also a 3D printer. But along the way, we were able to modify our undergraduate curriculum, which was revised to include the BBI 4206, which is microfluidics, biomemes, and bionames. So this was to introduce the innovators and especially the students to the field of microfluidics. However, we've been having challenges of having providing necessary practicals that the students can actually use, and then also a few limited prototyping resources around microfluidics to actually support our microfluidic course that we're offering at Makerere under the biomedical engineering programs. And then also, we really have very few collaborations around microfluidics, despite the fact that we've been able to actually do prototyping in all the other areas. And limitations are still there, especially for like microfluidics, as I've said, and then also around the lateral flow diagnostic innovations. And through the collaboration and also the different meetings and online exchanges with Professor Maiwin, one of our students, who was Edgar, was given an assignment or a task to actually come up with a microfluidic Y channel. So, and we wanted to see, it was a bit hard, okay, because we've never tried this, and we thought we couldn't take this on, although Edgar went to pursue her studies in Italy, and then we had to get another research assistant, who is Elvis Oroma, I think he's on the call, to actually talk on this assignment. So the assignment was to develop a Y channel, and then also to test whether it is actually working, whether we can have it from one end and then also can it flow through the other end. We were fortunate enough that we were having some acrylic sheets in the shipping container or the design space, but then also within town, we were able to actually be able to access a few acrylic sheets that we were using. And then the shipping container, we had access to adhesive transfer tape, but then also we also had access to chloroform that we actually used in our first bonding, okay, for the different plates that we're actually making. And then fabrication at Makerere, especially at the biomedical engineering unit, we don't have a laser cutter, so we had to go out in town, especially Elvis, to look for the shipping, to look for the laser cutter, okay, to come up with different dimensions. Although the challenge was more to access where can we have it, and then also seeing can they actually give us what we needed. So at first, we after going in town to actually fabricate and come up with this, we then had to look for how are we going to actually bond this. We were fortunate enough in the shipping container that we had chloroform. Okay, and the head of that shipping of the design space, Julius, was able to recommend use of chloroform. However, we didn't have a tight bonding with for the different plates when we used chloroform. And then we reported back to Prof. Maiwin, who recommended probably we try thermal bonding with the chloroform. And we had to look for a heat press in town. But before going for a heat press, we actually said, I think we can try bonding these two using a flat iron, which was to give us the temperatures that we were needed. And then we also had the ethanol that we are going to sandwich between the two plates to support the bonding. And we actually got a perfect bonding compared to use of chloroform. So, and after trying out this, we then had to go out and look for a heat press in town, which they used to put markings and labels on clothes, okay, for sports uniform and extra fabrics if they want to label them. So we also tried out that, and the bonding was actually still better than the one that we used for chloroform. Although in the design space, we actually had chloroform already there, and then also there was the adhesive transfer tape. So we've been able to actually try out and see that we can actually be able to fabricate this. Okay, however, there are still challenges that we need to navigate along the way, and the way forward for this is we want to explore other designs and also safer bonding and effective fluid handling techniques. It is a challenge to look for really small tubings of really small diameters. However, we've been able to identify a supplier in town who can avail us with those. And then also we've talked to the head of the designer workspace at Makerere to include microfluidic lab consumables in the next semester procurement plans, where we are going to actually be able to offer the microfluidic course. So we hope the students can be able to take on to do some practicals around microfluidics. And then also we want to identify potential diagnostic challenges that we feel could be addressed around microfluidics. And then also would like to map a current landscape around microfluidic design and fabrication. We want to see where are these resources distributed in the university. We want to see what's available in the other colleges, what's around in the other institutions in Uganda, and then also the region, and see, can we share probably design processes. And then we want also to build and strengthen the current networks and collaborations, for examtle, with Heriot-Watt University, through Professor Maywin. There's Professor Mark Demolier, there's University of Edinburgh, there's Mbara University, and so many others, which we want to continue building that collaboration and then also strengthen that. We'd also like to support evidence-based development of fit-for-purpose diagnostic innovations, so those that can actually address the needs of the needs in Uganda, in East Africa, and in Africa at large. And lastly, we want to go in for look for funding to actually support research and design and translation. So we at the biomedical engineering unit, we are trying to focus so much on design and translation, but then also alongside research. And with that, I would like to thank you all for listening to me, and I want to hand over to Ayo and Prof. Maywin.
Thank you very much, Brian, for sharing these first steps in microfluidic prototyping. So I don't know if some of the audience wants to ask a question. I think we have time for maybe one or two really quick questions. I think my question is, Brian, do you think that the current sort of setup where you have to laser cut in town I suppose at a maker lab, is suitable for your teaching activities, which start in January?
Okay, can I respond now?
Yes.
Okay, yeah. So there's the students go through a 4-year design course. Okay, and it is 4 semesters of design course, but also one of the assignments and tasks that they have to do is to actually look out. The university can't provide all the resources that they need to actually for them to prototype. So if we are able to identify the areas where the laser cutters are, I don't think we shall have challenges for even the students to be able to access, because currently in their design module, which they do over the 4 semesters, they're actually able to access some facilities, some resources and facilities in the university, but those that they can't, they're actually able to go out and access that. So I feel we can take on this, but then also we are trying to look for other avenues on where we can have the laser cutters be brought at the unit through writing different grants and having this part of them.
Thank you, Brian. And obviously, there you focused on acrylic, but then there's really cool things you can do in paper, which is much more sustainable. And there's plenty of people on the call, who could help with that as well. I don't know if there's any questions. Oh, there's a question from Jackie. Jackie, actually, are you able to turn your mic on and maybe ask the question live rather than me reading the question?
It was really just an appreciation for your talk. I love the creativity and getting the microfluidics done. I had just come across the Gates Grand Challenges Explorations funding sources, and they had one specifically on a call for innovations for women's and female-specific conditions. And with your preeclampsia test that you mentioned, you might be interested in applying for that. And then there's some other sexually transmitted infections that anybody on the call might be interested in developing. So totally just a comment, not a question, but an appreciation for your work.
Thank you so much,
Thank you,
Jackie. All right. I think in the interest of time, I'll hand over back to Ayo now. Thanks again for coming on the call, Brian.
Thank you, Brian.
Thanks
it's really cool work that you're doing and the creative solutions that you're coming up with. So we're going to move forward, and I'm just going to do a quick introduction for Dr. Manu Prakash. Many of you know about his work, but his lab and his work has demonstrated a dedication to inventing and distributing frugal science tools to democratize access to science, diagnostics for deadly diseases like malaria, convening global citizen science communities to tackle planetary scale environmental challenges like mosquito surveillance, plankton surveillance. And there's notable inventions from his lab, like the Foldscope and the Paperfuge. I was personally excited to see some of the work that Manu's doing across communities. For example, speaking at the annual meeting of the American Society for Cell Biology that's coming up in December, and really sort of work that's multidisciplinary and crossing communities. He's won several awards, as many of you know, including the NIH Director's New Innovator Award, the MacArthur Fellowship, and the Mueller Colworth Prize. Today, he's going to be talking to us about capacity building via community-driven science. I'm very excited to see what you will tell us. I will stop my sharing and hand over to you.
So first of all, thanks everyone for coming. And then I always have this question, which is, how much time do I have? Do we know? It's 9:25 on my end.
Yeah, we're
I want to
aiming
leave some time for discussion.
Yeah, so we're aiming for about 25 minutes, leave 5-10 minutes for questions.
Okay, that puts me in a tough position because I am always bad at coordinating time. So what we'll end up doing is I'll talk for 25-ish minutes, and then just let me know, and we will arbitrarily stop wherever we are and turn this much more into a discussion.
Sounds good.
And I think if any of you you want to interrupt me somewhere in the middle, I'm very happy to do this very informally. I think first of all, I want to start by just saying, I think the challenge that we have all taken on as a call of duty in our lives in some sense is something that requires this type of an approach to science. So I'm really delighted that many people are now coming together to build this. I know in the past, there have been attempts in that space, but frankly, it's frustrating to see how slow change has been. So, although technology is a very powerful tool, we all have to be, in my mind, a lot more hungrier in some sense in making the changes that we do want to see happen at the grounds level. And although change has happened, change improvements in healthcare across, I'm just looking at the 10 years of my own career. I started the lab in 2011, and I see dramatic changes. But frankly, one thing that I deeply believe in is the fact that in order to be able to expedite and bring solutions to people faster, we will have to take approaches that are different. And if COVID taught us anything, it really did teach us a remarkable strength in scientific networks that were created. So I think, this is just a call and almost a thank you to everybody that's been putting this network together, people that are spending the time. Try to recruit as many people as you can, because I very strongly believe that without a community approach, I don't think we'll be where we want to be. And what's the point if 10 years from now we reach and we realize that we haven't actually enabled the solutions that we all cared about? Okay, so with just that as a rant, let me tell you a few things that I've been thinking about. Few examples and case studies of things that have scaled up and how and why they scaled up, lessons. If I was to do it again, how would I do it? So first of all, there is a very broad range of tools that we've been building. I won't have time to talk about much of it. So I will spend less time on the technology. Much of that is published openly. A lot of tools that we make are open access, open hardware. So you all can dig into the details on that. It's quite a broad portfolio at this point. And I think my biggest challenge when I'm thinking about these sets of ideas in the context of frugal science is start thinking about scale from day one. So any project, any set of an idea, we truly are thinking about a cost performance framework, and scale really comes from cost. And so it's very important to be thinking about that early on. Another philosophical thing that I want to start on is, as technologists, and I know many people in this group will associate themselves with technologists. I have a friend in Sudan who is taking my class. I teach a class called Frugal Science, And we were having a conversation just a week before the war broke out in Sudan. And he told me this Sudanese saying that some of you might know, and I really take it to heart. What he said is that there is a saying in Sudanese, the pen that does not eradicate ignorance and shame. And what he meant by that is the pen is a symbol of technology. Let's make sure that we make and build technology in the world that truly brings the change that's needed in the world. And so technology for technology's sake becomes very difficult otherwise, because it might not eradicate the ignorance and shame that you might be after to begin with. So I think symbolically, this is to me one of the goals of building healthcare technologies, is making sure that they reach in the hands of people that we care about. I think although all of you are focused in health in some sense, I just want to mention that education is an incredibly important and integral part of health systems. And so any sets of spillover in the context of bringing the capacity to create these solutions at a larger scale is incredibly important. I don't have to tell you about the challenges in the context of health, but I think the other third pillar of much of this work is the environmental side of the story. A lot of diagnostic tools that we've been building have found homes also in environmental monitoring. And sometimes, for example, a disease that we work on in schistosomiasis, we've been grappling with this idea is that no matter how much diagnostics and how much therapeutics was put on the table, unless an environmental factors and environmental innovation happened in being able to monitor these diseases, we will have a very difficult time in eradicating. And of course, some of this is also related to the depletion of many of our environments that we care about. So let me begin by just saying this is a large challenge that we all have been thinking about. And to a certain extent, in terms of the tools that we make personally in the lab, the audience always happens to be community health workers. And one of the challenges that we have to be thinking about is that the tools and technologies we build will actually operate and work in hands of individuals, and who I might, for lack of a better term, might call amateur scientists that have certain amount of training, but not as much as you might think of a lab technician, for example. So a lot of community health workers that we work with in fairly remote parts of the world would qualify in this scenario, that they have spent some dedicated time getting trained, but don't have a formal education all the way to a lab technician, for example. So we have to be thinking about whether these technologies will work in hands of people that we care about. So I'm going to now transition and just share a few sets of tools. I don't think I'll have time to share everything under the sun, but as I said, a lot of it is published, and I'm happy to have sets of discussions. Okay, so let's start with manufacturing. This is something that is incredibly important in thinking about building capacity. This was a COVID context for us when we started looking at medical devices, and we asked ourselves a question: could we truly take a car manufacturer, an entity that was building and manufacturing cars, train them through the entire process of medical device design, risk management, regulatory processes, and build a high-end ICU ventilator, for example? We chose a difficult project, and this was a large effort, 60 people involved, 3 countries, 3 manufacturers from India, Nepal, and Kenya. What was common between our partners on the ground was they had phenomenal manufacturing capacity, but no life science and biomedical capacity. So this is a lesson that we learned. By the end, as you can see in this video that I just played on the left, you can actually see a complete ICU ventilator manufactured, tested, and gone through the types of clinical validation in India, Kenya, and Nepal. Certain parts were built in 3 different places. And this was a phenomenal experience for everybody involved to learn the process of what it takes to make high quality instruments that are going to go through regulatory approvals. Some amount of manufacturing capacity was necessary to be present as well. So it's valuable as a lesson to not start from zero. But what we ended up doing was looking for industries that had some of that capacity and pivoting them in building and designing medical devices. So this is an ongoing experiment. We're kind of documenting this as a case study. There are lots of lessons here in terms of building training programs around medical device design. And I'm happy to talk about this, but I just wanted to share this just as a single slide that starting with manufacturing might actually be a good idea in terms of what is lacking sometimes in places and challenges that you all might want to focus on. I think moving to kind of more point of care diagnostics, I have to share a personal story. We've spent a lot of time in the field. Often enough, the problems are motivated by our conversations in the field. And I have a colleague in Uganda who had been a pathologist for the longest period of time. And 10 years ago, I had a conversation with him out in the field. We were running our malaria trial, which I'll show you what have we done over the last 10 years. And what I found fascinating is he told me to go back home and he said, Manu, come back to me when you can do diagnostics under a tree. And, the first time he said it, I didn't really understand what he meant, but I know what he meant later on. Really, diagnostics capacity that's untethered to any ecosystem, not on a hospital bench, not on electricity, not on a requirement of fresh water and other things. So, that's what I've taken to heart. And a few sets of tools that I'm about to show you are really inspired by that philosophy of can we do even, for example, molecular and high specificity sensitivity diagnostics under a tree. So I'll show you one example. Again, this is something that we've been working on for the last 7 years. But COVID accelerated much of this work. And, I don't have to tell you all this, that even with the advent of a massive growth and increase in technological capacity of the world, so 3.2 or so billion tests were performed until 2021, end of 2021. Less than 0.4% were in the context of LMIC, and that is primarily driven by the cost framework, because many of these, neither the distribution nor the cost for a large number of tests made it to the grassroots in some sense. We've been also thinking quite a lot about the environmental cost of diagnostic tests. And so I think this is something that made us rethink about role of electronics, especially in disposable one-off tests. And I'm thinking quite a lot about types of tests that are driven in the context of cartridges that are associated with this. This is another lesson to keep in mind. As we invent these new sets of technologies, we have a chance to really think about environmental footprint as well, although it's a very difficult challenge to think about. So let me just share with you one quick project that's not published. We are about to submit this. This should be online in a couple of weeks. But one of the threads is, how do you do diagnostics with no infrastructure, so no electricity, but still be able to do molecular diagnostics in the field? Very much like an LFA test, could we really do something like a LAMP assay, but with minimal sets of resources? And that led to, of course, we did this for COVID, but I want to remind you all that COVID is just an excuse in some sense. We just spent some time in Liberia. We're looking at Lassa fever, for example, and I'm happy to talk a little bit more about the directions that this tool is going. But the data that you're about to see will be in the context of COVID. And the idea is fairly simple. I think many of you have been thinking about this in the context of Reclone, in the context of many other devices. And the framework is, could we demonstrate manufacturing of all the reagents all the gadgetry and distribution in a manner that requires a fairly small footprint to be able to manufacture a test literally anywhere in the world. That's the goal. I think we are, I would say, 70-75% there. We are making progress, and I'll show you in the very end where we are going with something like this. But just to give you a context of the device, this is a device that we built. We call it SnapDX. It's sort of a Russian doll. It has lots of chambers. It kind of is designed like a ballpoint pen. And what the device does is take samples. It can take saliva, blood, urine, sputum as the 4 samples we've designed it for. We have the capacity to do lysis, extraction, and then amplification and readout without electricity. And in some sense, the way you should think about this, that the cartridge and the gadget is one and the same thing. This thing depends on hot water. There's lots of sort of steps involved in terms of what internally happens in the device. But to the user, they essentially push a plunger from one side to the other. Actually, a lesson that we learned in designing this device was actually it took us more time to design the IFU than it took us to design the actual device. We ran, to prove to ourselves, we built 10,000 of these. We ran 2 clinical validation studies and usability studies, 5,000 people in the Bay Area, and now another one that's running in Guatemala. And one of the big things that ends up happening is the iterative process in design is incredibly important. And no matter how beautiful a device you make, it has to work in the hands of a person who has never used something like this before. So that was actually a really wonderful teaching moment for us itself to be really thinking about IFU design. We've been making all reagents from scratch. There is lots of platforms for LAMP now, including many reclone. We have compared many of these sets of enzymes, and I'm happy to share what works and what doesn't work. But I think one thing that I do want to share is it was important for us to build a manufacturing capacity for this. That's why we decided to make 10,000 of these to prove to ourselves what the cost of this gadget would be. And currently, everything, including reagents and the instrument, is around 84 cents in parts. Of course, it requires a lot of manufacturing tools and ticks to be able to do something like this, but that's really where we are currently. And I think, there is a validation study that we are just publishing in that context. And I think the big thing that I want to show you is there are still things to be improved on the specificity sensitivity for COVID. We are roughly between 95 and 82%. There's a lot of work that we can improve on this. And one of this really comes down to the thermal side of the process. This uses hot water. But kind of the direction that we are taking this project is to now, once we have demonstrated that we can make this, we are putting together essentially 2 shipping containers that have everything from scratch, all reagents, all enzymes built from scratch, all gadgetry made from scratch. We've identified 2 countries currently where we are deploying this to test an idea. Could an ISO grade facility for making a molecular diagnostics test could be done at scale? If any of you are interested in engaging, because I know many of you are in places that are interested and excited about manufacturing, I would love to engage and see how quickly we can expand the capacity and explore many other tests. I think one of the things about this is we are broadening the number of tests we want to run on a platform like this. And again, it's designed around saliva, sputum, urine, and blood. I'm going to switch to another tool, which is sort of interesting to think about as a platform around centrifugation and sort of what happens when you share openly ideas and design to a broader group of people. This is a story that's on the centrifugation side. Some of you already know this story. This started from 2013. And one of the challenges that we had identified was to really do high-end centrifugation in the middle of nowhere without electricity. And one of the flights coming back, I was thinking about toys. And of course, yo-yos came to mind. I had a few students in the lab who were really amazing yo-yo player. So I'll just show you what Dino happened to be a circus artist. And at that time, he actually held a world record. Now the world record is broken, but yo-yo you can throw roughly around 4,000, 5,000 RPM. Clearly fast enough, but quite dangerous sometimes when you throw these things around. But in that process, we ended up stumbling upon this toy, which is called a button string. Okay. And one thing to think about is this is an object on the right. Many of you might have played with this. It's a fairly simple object. And the surprising part about this, and I think all I'm trying to do is identify the role of play. Sometimes in the act of, working on difficult and hard problems, we have to be thinking about play. What led with this scenario is when we looked at this toy it's the oldest toy in the history of mankind. It's the 5,000 or so years ago, people have made these sets of toys, and in all cultures around the world, people have used these toys. Unfortunately, nobody knew how it works. So we asked that question. This is how it works. And again, the reason I want to put an equation in this slide deck is the appreciation that when you push the bounds of engineering, you're able to do things that you might not actually even appreciate with very simple parts. What we were able to do when we understood this tool is to break the world record for the fastest spinning object now with human power. That's roughly around 125,000 RPM. So this is equivalent to around 30,000 G-forces. In a couple of minutes, 90 seconds, we can separate plasma from blood with a tool like this. We can pull out single malaria parasite, for example. And one of the fun things about something like this is because it's not a black box, many of the communities that engage are excited about building on top of it. People understand the tool. And this is another lesson to take, is going away from building and designing black boxes to really designing tools that people can appreciate and build trust around. What was fun is when we shared this design, hundreds of designs started popping up. I know many of you work on centrifugal microfluidics. Any of you can take these sets of frameworks and ideas and build on top of it. And actually, there is a UTI test now running by fidget spinners. I know there is also an amplification assay a couple of people have built on the same set of a platform and an idea. But it allows us now to do high resolution centrifugation with no electricity. During the COVID time, we built a test around a framework that we call HandyFuge. Now, this is a similar framework, but essentially building something that's a little more robust and also for larger volumes. Again, a fairly simple framework, but it now allows us to implement any kind of electricity-free centrifugation in a field in a very portable setting. All the design and part files for these are openly available. So if any of you who want to build these sets of things can essentially build on top of these sets of platforms. So the lesson here is sharing platform technologies, because there are far many number of problems, and sometimes other people will build upon the tools that you invent. And it's actually quite a powerful ecosystem. And I'm going to, maybe for the last 5 minutes, I'm going to transition to tell you about a little bit on malaria and microscopy. But before I say this, I think one important thing to understand is how essential some of the microscopy and microscopy-based tools are. A friend of mine did this playing with ChatGPT. The question they asked ChatGPT is, what are the top 5 most important tools for science? And the number 2 tool that ChatGPT thinks is a microscope. I know many of you won't disagree with it. It's something that allows us to see the invisible world. It has a tremendous power, but I think it has not made the kind of impact as yet that it can in a primary healthcare settings. So I'm going to spend a little bit of time talking about that. There is a link between basic science and applied science. So to a certain extent, a lot of clinical work also depends on microscopy. And we've been thinking about this link to make sure that we make these tools much more broadly accessible to researchers as well as to healthcare partners. So with that, we started thinking about microscopes. I mean, of course, you all know Foldscope as a story. I'll just say a word or two about it in the very end. But the three other tools that we have recently released in the last 4 or 5 years, one is a planktoscope. It is for aquatic ecosystems. And the other one is Octopi, which is an automated malaria diagnostics platform, and Cephla, which is a platform for doing a very large number of assays in an autonomous fashion. So let me give you kind of a framework for why we did this. This is the total number of tools that are out there. So I'm only going to spend time on 1 or 2 of them. But just if you go read some of the papers around squid and octopi, you'll find a very broad framework for building automated open source life science instrumentation. So just backstory, why malaria? This is one of our field sites in Kalahandi. And what you're seeing are 2 of my colleagues. They spend 10 to 12 hours sometimes in the clinic just diagnosing malaria. The current WHO standards are based on a Giemsa stain that was first described in 1904. So this is sort of depresses me sometimes that innovation, although moves fast in certain frontiers. And the irony is we have automated cars on our streets driving around. And while we still don't really have many automated platforms on the imaging side. So, long story short, we started building this framework. It is important to understand that RDTs alone are not sufficient for the challenge. One of the things that you've all heard about is the HRP2 gene deletions have started to and been detected all across the world. And that leads us in a very vulnerable position currently for what RDTs cannot actually enable. So this was the evolution of this hardware that I'm about to tell you, all the way from 2015, 2016, when we built the first instrument, now to the final instrument that I'll show you. This is an instrument that takes a glass slide and tells you in real time whether you have malaria or not. So the instrument has been growing in some way. We use a machine learning framework to do this entire framework. And then just give you the context of data. This is a data set collected by a healthcare worker in Tanzania. And you're looking at an entire slide. And within 5 minutes, the system is capable of collecting data for around 20 million cells. So compared to a human that essentially looks at around 3,000 to 5,000 cells, now the system is looking at orders of magnitude higher. And one of the powers of that is that allows us to do the kind of performance that in real time, the system for 25 parasites per microliter, we can do 100% sensitivity and specificity. It's not as good as a molecular test, but I very strongly believe that imaging will give the run for the money to get better and better as we get better in the context of thinking about clinical validation. There was a problem. Oyo, am I out of time?
Oh yeah, maybe we can wrap up. So I see many questions in the chat. So maybe
okay, I'm just going to say maybe 2 words about this. Sometimes when we think of an idea, we get too excited about it and then we hit roadblocks. We hit this roadblock on this work in India where we started seeing that the quality of the slides was not good enough. And then we went back and we looked at what was happening, and most slides are actually made in the field. And for that, we had to go back and invent some of the most simplest thing that you all know and do, which is how to make a blood smear. It actually turns out it's not trivial to make a blood smear. And some people get good at it, and some people are terrible at it. We had to ask ourselves the physics of how to make smears, and that led to this device. Again, just using 17 cents of a syringe, 3D printed part. This is now an instrument that's completely open, accessible to anybody. Any of you are interested can go on GitHub, print this part. This makes essentially perfect slides. And now we're quite excited because it allows lots of people to be able to make blood smears for all kinds of things. And again, we are using this tool to understand how 3D printing can enable formation of or distribution of medical tools. So I have to show this video to just give you an idea of 10 years that it took and where we are today. At this point, Octopi can, if I just put a slide in there, it has the capacity in real time, essentially identify pathogens. We started sharing this tool with researchers. Now 10 of these machines are out in the world. We're trying to get 25 or so of these machines. The instruments essentially do autofocus. And then once we start the process, you will see while it's scanning, it's doing real-time ML to be able to identify parasites. So now the scanning has begun. And one thing that I'm really excited about this that I want to share with this community is we are releasing this as a platform. So if any of you are interested in other diseases, we shared this with a group in Nepal, and within 9 months, they wrote an app for it that now essentially enables sickle cell diagnostics. So what's important here is to note that while it's scanning, it's actually picking up parasites. So there is no telemedicine, all powered by essentially a battery that's inside this box. And what I'm excited about is a lot more number of people engaging and writing and building apps for a platform like this, because then you don't have to build everything from scratch. And we can essentially, and again, this entire hardware is completely open. We're engaging with a lot of people. I'm going to close here. I think, unfortunately, I don't have time to tell you about a lot of other things that I could. But we are short of time. I'll end with, Foldscope as an anchor point in terms of thinking about tools and technologies. This is a tool that some of you might have used. It's been 10 years we released this tool. There is roughly around 2.1 million users around the world currently. And they do remarkable things. There is roughly around 500 to 600 papers written on all the way from human health, animal health, agriculture, education. And there is a single reason why we were able to scale this tool, which was that we started with a community-first approach. We distributed the tool to 100,000 people from the lab, no questions asked. They could do whatever they want with it. They didn't need to report to us. They didn't need to tell us anything. It was good if they told us what they were doing, but other than that, they were free. And we focused on manufacturing to make sure that we could deliver and get these tools for $1.75, which ended up being the price that they are currently available today, to match and mirror a price point that you can see a single bacteria. So, sometimes in scale in markets that are very price sensitive, it's quite hard. So you have to create a kind of a community and a class of users that will build applications on top of it. It was quite difficult in the lab with no support to actually manufacture 100,000 microscopes. That was 2 students and me, but we took on that challenge because we believed that seeding a community is far more important than the technology itself. So, this is sort of a lesson that I carry on with a lot of work that we do, that sometimes it's in the communities that drives much of this. And, this is really what's powerful, that other people are able to build upon the work that you are excited about once you share those sets of tools. So, Nigeria, for example, has 5 distributors of Foldscope currently, and I kind of get a lot of joy out of it because suddenly people have been able to build businesses on top of it. There is a community platform where people can document what they do, and much of this is written. So I'll end with this picture, and I can take questions. This is a picture from 2 days ago, and pictures like these come out every day. As people are starting to engage in their process of science or their own journey. I'm excited to say we are building a manufacturing plant in India, and then the next manufacturing plant will be in Africa, and I'm trying to figure out which country it's going to be. So if people have suggestions, ideas, interest, do let me know. I'll stop here. I'm sorry I took much longer. I wish we had more time. I would tell you about many other things. And then maybe as a closing, I teach a class called Frugal Science. It starts on January 5th. It's open, free to anybody anybody who wants to take it. So just if you search for frugalscience.org, you can find that class.
Thank you so much, Manu, for an inspiring talk. We'll definitely have to do an encore sometime so that we can hear more from you in the future. I know there have been many questions in the chat. We can keep this running a little bit. I know some people have to go. We'll summarize the Q&A as well in our blog post. But yeah, I'll just turn it over to questions. So feel free to use the raise hands function. I also saw a few. I know Jackie had a question earlier. I know Chang Hee Kim had a question earlier. So
yeah,
folks...
I haven't seen the chat. So if anybody can
yeah.
questions, it helps me because you guys can then prioritize.
So I see Jackie had a question earlier, and she talked about, the IFU design, and she asked if you saw different needs in the instruction designs in the US versus Guatemala.
Absolutely. I mean, it was not a translation of language. It was a complete redesign of objects people were used to. And we really heavily relied on the idea that everybody knows how to press on a syringe, but there are still nuances, and it was an extremely iterative process. I think, to be very honest, we designed the IFU more than 30, 40 times. And it was a learning lesson where we would videotape people of how they're interacting with the device, go back, change it, because one of the goals is that, of course, we could have them watch a video, but many a times that might not be feasible and possible. And so we had to rely on a paper. I think videos are clearly much better instructions. And some of the community has a cell phone. So we took the IFU, broke it down, and made an app for it so people could interactively go through the IFU, and some steps were in video rather than in figure. But because of the sets of requirements around regulatory requirements, it became important to also have a paper-based system. But just something to mention to people, if you're designing a diagnostic test, keep that in mind, because if somebody makes a mistake, that counts against you in terms of sensitivity and specificity. It was not that the device failed, it was just that the step was not performed as you might have desired.
Thank you. That's a very detailed response. And I see Jackie has a response saying that's a really great case study, and she'd love to learn more and read more about that experience. Julius has their hand raised. You want to go ahead, Julius?
Yes, I thank Dr. Manu Prakash. I think I have actually been in contact with Dr. Manu, and I've attended some of his classes, and he's really very impressive, and his openness and his way of doing things. But he's talked about setting up things in the container. And I don't know where these facilities are going to be. So that's why I was saying in the chat, have you figured out where these facilities, such a facility will be set up and which conditions? Because at least we have lessons for now about 2 years or more. We've been running this makerspace in design in a shipping containers repurposed. And I think I showed you a tour one time. So we'll be willing to share lessons and some of the things we've learned setting up such things and the environmental conditions and some of the inputs you have to do.
Yeah, so Julius, that would be fantastic. If you can send me a message, we are very open about, I think funding is one of our only limitations currently. The first one we have committed to setting up in Senegal, but beyond that, I am looking for places. I know Cameroon is pretty high on our list, and it would be phenomenal to just have a chat. And I think one of the goals is, shipping container is just as a iconic thing. As long as there is any space, the goal is to demonstrate an idea that a small footprint is sufficient to build high quality devices. And so I think this is kind of that trade-off. But I think the whole point of this network is to be able to engage with each other. So if you have any interest in that space, I would love to chat.
Yes, I will have to. I have your WhatsApp, and I think I'll be able, because the landscape in Africa and the temperatures are almost the same. So I would tell you some of the things the mistakes we made in our shipping container maker space until when we got to a feasible one.
Yes, we definitely need to learn on each other's mistakes. Chang, I think your hand is up.
Yes, thank you for your wonderful talk. So how do you get your diagnostic test that are non-laboratory diagnostic tests, how do you get that approved for non-lab use when there are so many steps, like human intervention steps?
Yeah, I mean, I think one of the threads is that it's the usability and the validation studies that get the approval. We did, 2 rounds of usability studies with people that had never seen the test, had never used it, are not scientists. It was just an open recruitment call, and anybody could sign up. We measured the demographics, and we essentially publish everything along the way. And that's really sort of the framework for thinking about approvals. This is also why it's important to have the capacity. We made 10,000 tests primarily because we wanted to understand our quality control. We wanted to have enough failure points, a large enough cohort to understand. One failure point that had come about at certain point of time was color blindness. This has not been really highlighted enough. We had to change our design and our test to then change for common conditions like color blindness that do exist in a significant population. So it's an iterative process, and you just have to do it at a large enough scale. I think the philosophy that at least we apply in our work is, although we start as an academic lab, I mean, we are an academic lab, we are focused on making products. And I think that has a different mindset. So we can publish a given result for a certain amount, but to transition and turn it into a product really does require a longer term horizon and a longer term effort, and to be able to scale that efforts. And
Yes.
This is also where the network becomes very valuable, because many a times you will find you would need resources that are far outside of what you might have access to. Like, we have regulatory consultants in our team that we just hire for consulting that is quite expensive. And if this network had a few regulatory consultants that would be willing to work pro bono early enough, it would be phenomenal for many projects, because they could tell you early on some things that we should or should not do.
Yeah, so what regulatory approval are you seeking?
I think right now we are essentially doing sort of 2 sets of things. One is at-home test. We are submitting this in the US itself. And I think the other team that we are talking to on the regulatory side is in India. And that one is really designed much for a community health worker, not an at-home test. So those are 2 different streams, and then they have different requirements. And I think there is a couple of conversations which would be valuable to have. I know that post-COVID, there is an international framework that's growing for regulatory approvals across multiple countries. And we haven't engaged in that process per se as yet, but I do know that there are efforts right now associated with going away just purely from a FDA and a CE marking. But currently, we are just following FDA framework.
Thank you very much.
Thank you. Thank you so much. Thank you, Chang-hee. Thank you, Manu. We have time for maybe one more question, and then we have to wrap up. We are very much over time, but it's great to see the enthusiasm and the question.
Yeah, and I'm happy to stay on the call. So if people have to leave, I totally understand. But if anybody has any questions, I'm happy to chat.
Okay, so maybe we'll just close formally, and then we can open it up in case people have questions for you. So I'll just very, very quickly talk about what's coming next in the network. So we have an in-person workshop December 7th to 11th at Ashesi University in Ghana. Really, our hope is for this to provide an opportunity for hands-on demos, group discussions, co-creation sessions to address regional needs. This will be a small workshop because of cost and time. We hope to see some of you there. But really what we're hoping for is that this will kind of seed other in-person workshops and other in-person meetings. So please reach out if you have ideas for places we can meet, if you have ideas for things that you would love to see, and we hope to see many of you there. Then early next year, which is strange to say, but early 2024, we'll do our next online meet and greet. So please reach out about that as well. These are some of the events that we highlighted. We've got that Bioengineering for Global Health workshop conference in Nashville, the 6th Africa International Biomedical and Biotechnology Conference in Kenya, and the African Society for Laboratory Medicine meeting in that's going to be in Cape Town. We'll post all of these on the Slack. We'll send updates, and please feel free to share other events that you're aware of. Please stay in touch. Please connect with us. If you have feedback, suggestions, messages, you'd like to highlight your work, you can reach us on many, many different places. We'll have a blog post, hopefully, that will go up about this one, that will have the recording. Please share with others that might be involved. We want to thank you all for your time, attention, patience, enthusiasm, engagement. We hope to see more of you soon. And then I'll leave this room sort of open informally in case folks have more questions or feedback for Manu. Thank you so much, Manu. Thank you, Brian, for talking to us, for inspiring us, for showing us what you do. I'm very grateful for all of you.